Thanks, Roy, for that really excellent presentation. And I must say this last slide is a little different from your other slides. It's a big font, one sentence versus a fairly dense presentation. It was really excellent. So there's an academic view and a summary of monotherapy and then some discussion on combinations. But one of the two sort of areas that are really perhaps lacking at the moment is understanding how to select preclinically combinations and then how to prioritize combination therapies as you move into early stage drug development. So for this next section, we have 4 folks, 2 from academia and 2 from industry, and looking forward to their presentations. First up will be my co-chair, Samir Khleif. And Samir is the director of the Loop Immuno-Oncology Lab and the biomedical scholar and professor of oncology at Georgetown University's Lombardi Comprehensive Cancer Center. So, Samir.
Thanks,
please.
Roger. So I'm going to actually do a good job like Roy, but try probably to go through 70 slides rather than 100 in 15 minutes. You've done phenomenal, Roy, I must say. So the question is, again, we are going through multiple potential combinations with the checkpoints, particularly anti-PD-1, and those combinations are a lot. PD-1 has been used. You've heard about this now. I'm not going to go through details. You've seen this from Rami. There is 164 targeted agents for PD-1, PD-L1, and all of them are being developed either preclinically or now through phase 1 to approved. Combination immunotherapy, same source from Tang and Shalaby, CIR. There's 1,500, at least by the end of last year, a combination with PD-1, around 1,100, and the multiple agents are being combined with each other. We have seen in late ASCO multiple failures now that are not going forward. And the question is, can we select in a better way? And Roy mentioned that just before. It's not the number of clinical trial, are they scientifically guided? And are we doing the proper scientific due diligence before we move forward with those scientific clinical trials? So the question is, again, you've seen a lot of responses. The question is not the greens, but the upper ones, and how we can move the field forward by combining with PD-1 or PD-L1 and change the resistance into responders and enhance its effect. So immune therapy resistance could be primary or secondary, as we all know. So when we think of immune therapy resistance, there is general mechanisms, particularly including intrinsic immune biology, like lack of antigen presentation through antigen expression or processing, or T-cell deprived environment, such as in beta-catenin or MAP kinase, etc. And then there is a suppressive microenvironment that we all know about, and we have seen more and more of those mechanisms coming up that may lead to resistance. So when we think of combination, can we really think through those kind of system biology and take them forward? The other resistance, or what lead to the resistance, is treatment-specific mechanisms, such like low PD-L1 in specific diseases and specific organs or JAK2 mutations. But the other two that I would like to add here is one, immunocombination incompatibility, which means do immune therapies really work together as they're supposed to work together? Is it 1 + 1 = 2, which is synergistic, or is it possible that 1 + 1 = 0? And we need to start thinking of those in those kind of terms. And the second one, immune therapy biology incompatibility, which means when you use 1 or X, are you changing the microenvironment to a degree that then the Y is not going to be working the same? And these are the main 2 elements that we really need to think about when we start thinking of combination and how we do combinations. When we talk about combinations, there's so many. You can combine vaccine with PD-1, other immune modulators, standard therapy like chemo, radiation, small molecules, CARs, etc. 940 different IO agents in clinical stages. So there are many potential clinical combination. So how do we scientifically guide those type of combinations? I gave a talk at Karolinska a few months ago, and the title of the talk is Immunotherapy is not logic, combination immunotherapy is not logic, it's science, because we're dealing a lot with logic. We have this, we have this. We can combine them. Yeah, we can combine them. One negative, one positive. So is this true? So I'm going to give you today a couple of examples on those potential resistance that I mentioned. One of them, which is already published, but it's a PD-1 OX40 combination. It's clear, PD-1 inhibits the negative regulation. OX40 activates T cell. So clearly, if you combine them, you should work. And large clinical trials started utilizing those in the clinic. So we've done this in mice, of course, the experiments in mice where we combine anti-OX40 and PD-1. And interestingly enough, when we combine the anti-OX40 and PD-1, what we found is this is what happened with anti-OX40. You add the PD-1, the response is gone. So just adding PD-1 to OX40, it really eliminates the response of the OX40. When we look, which is this, when we look to survival, it's exactly the same thing. This is what happened when we give anti-OX40. This is what happened when we give anti-PD-1. And when we add anti-PD-1 to OX40, the response is gone. So in a way, there must be some sort of incompatibility between those two, that they really eliminate each other to the degree that I'm not sure that it would be helpful to be combined. So when we started looking and digging into the mechanistics for that, one is we found that when we combine them together, CD4 decreases in the microenvironment. And as importantly, CD8, particularly antigen-specific CD8, is almost gone from the microenvironment. So we just adding, this is OX40, adding PD-1 to OX40, it eliminates to a large degree those antigen-specific CD8. Now, when we look into CD8 to Tregs, again, the therapeutic ratio is completely gone. So there is a reason that this could potentially fail if we take into clinical trial. And then when we started looking into the reasons that this is happening, it's clearly not the activation of cells. Because when we look into the periphery and we look into those cells in the periphery, when we combine anti-OX40 and PD-1, the cells is specific against antigens, very highly activated, and they're circulating. The moment they get there into the microenvironment, they're dying. And they are gone from the microenvironment. So it's not the activation itself, it's what happened after the activation. So we also need to really understand, when we look into the science of those combinations, what we need to think about when we decide on this. So when we take this to in vitro and we decided to look into how this combination in the PMEL model lead to the elimination of that response, we started looking into potential death or potential apoptosis. So when we treated with PD-1 and PD-1, or OX40, or OX40 then PD-1, etc., we see a little bit of apoptosis. The moment we added PD-1 together with OX40, this is what happened. So the cells got absolutely berserked and popped up. And this happens even if we put OX40 afterward. So what happened is we are activating those cells to a degree that we're driving them completely into suicide, into apoptosis. And accordingly, they're gone. So if you look at procaspase or BCL-XL, it's the same thing. So I'm just giving you a flavor of this, at least for us to think about this. And when we look into clonality and fractionation, they're completely lost within the microenvironment. And this was done in collaboration with Adaptive. So when we think about it, a couple of clinical trials that were done so far did not show any positive response. At least the large ones that are being conducted. So when we combine them, it did not work. So there is a reason why they might not be working, and that reason is biologically irrelevant. So that's one example that I wanted to show you. The other example is how we also combine other things with PD-1 to make sure that we can enhance PD-1. Lots of people are working on vaccines now to try to enhance these T cells, making sure that we have more of those specific T cells that would be within the microenvironment. So when we block the PD-1, PD-L1, we have better effect. So the first one's published. This one just submitted, but I'll show you very fast data to show this. So when we started thinking, great, can we enhance PD-1 with vaccine? Because lots of people are thinking of neoantigens, combining them, and even with priming agents like chemotherapy or radiation therapy, what would be the best way of doing that? So when we combine the vaccine with PD-1 and we give them at the same time, we had pretty good response. Now, we did the same thing. We kept this exact same schedule, and we added 1 extra dose of PD-1 prior to that. And the response is gone. So the moment we block PD-1 prior to activation of the antigen or proper priming of T cells, those PD-1s lead to complete loss of response. And we've done them in multiple times, multiple formats, and this is the response when we give them together. This is the response when we block prior to that. Now, there is a reason to that, and that reason is when we look into the microenvironment, as soon as you do that blocking before, your CD8 goes down, and this is the vaccine with it together, so you have enhancement. However, when you block it before that, you go down, and specifically, the antigen-specific cells go down within the microenvironment. So when we further went back earlier to see what happened directly in early stages, we found exactly the same thing. So you can see that the antigen-specific T cells are completely or as low as if there was no vaccine when that blockage happened before. So when we looked at the status of those, they are annexin positive, and they're really dying within the microenvironment. And as importantly, if we look at activation, because the example that I showed you before, we overactivated when we gave OX40 and PD-1, led to apoptosis, and this is why the treatment is failing. In this case, we looked at activation. As a matter of fact, the cells are completely inactivated. So the reason they're apoptosing, not because of activation, something else is happening that is leading to apoptosis of those cells and disappearance of the specific cells from the microenvironment when we give that particular sequence. And accordingly, we need to take a look into it and make sure that why this is happening. Same thing, interferon, you see it's almost the same as if you did not give the vaccine. With that in mind, we lost also the memory cells from the microenvironment, and the model that we put together, which we tested, and it turned out to be accurate, is that when we're blocking before, we are actually depleting the further signaling of the T cell towards activation, and this is leading to development of dysfunctional CD8. We've characterized some of those CD8, and we found that if we do block beforehand, there is an increase in dysfunctional CD8 within the microenvironment, and the antigen-specific CD8 dysfunctionality is also increased tremendously with very low activation of those cells. I mean, you can see they're almost not there. So they're very dysfunctional. They're just sitting around and change their mind. They're not even being able to be activated with a proper priming afterward. So neither by interferon gamma nor by the CD40 ligand. And as I showed before, they are highly apoptosed and they're gone. So we block it, they're very inactive, dysfunctional, and they die. So the bottom line is that this is exactly what happened within the model that I showed you. So then we went back and we said, okay, if this is the case, then is this treatment in a resistant model doing the same thing by itself, which is the PD-1? And the answer to that was yes. It induces high amount of dysfunctional CD8 that are completely apoptosed. So we thought we'll go back and look into the clinic. So we looked into those phenotypes within the patients in collaboration with both Jed Wolchok and Nir Hacohen, one from clinical trial at Memorial and one in Boston. And interestingly enough, when we looked into those patients that were treated PD-1 or in combination, we found post-treatment that clearly 100% of those patients that failed have that in their microenvironment, and this is tumor. And only 25% of patients that did not fail had them, and you can see the difference here. So did we say that could we actually use this as a biomarker? Can we predict whether that is the case or not? And the answer to that is yes, it's pretty strong. 80% of patients that will not respond will have it in their microenvironment before of more than 10%, and only 22% of patients will have it if they're responders. So that's pretty strong. The question, can we also see it in the periphery? And can we follow the patient? And with Jed Wolchok, we looked at some of those, and the answer was very, very striking, that when we looked at the non-responders versus the responders, all the responders have their cells in the periphery went down. Most of the non-responders went up, and we're talking about 6 to 9 weeks post-treatment. and accordingly, we probably be able to predict after starting even the treatment who will be responding potentially and who will not be responding. And the AUC of those is almost 0.85, so it's pretty high. So I hope that I showed you at least a couple of examples when we start thinking of combination to enhance or reverse resistance, that science is very important and will guide us toward what we need to do and how we need to do it, and how to move forward toward putting together those combinations and why. Acknowledge all the people that work in my lab and the people we collaborate with, and of course Jed and Nir for their help on this. And oops, I don't know how to go back. Okay, Jed and Nir for their help on this, and thank you very much.